In this lesson, we'll take a look at the support cells of the nervous system known as neuroglia. We also call them glia or glial cells, Glial. So the job of the neuroglia really is to support and to protect the neurons. Again, think of them as really support cells. Look at this. They make up about half of the volume of the nervous system. And we have a total of 6 different types. 4 of them are in the central nervous system, while the other 2 are in the peripheral nervous system. So in the central nervous system, just as an overview, we have astrocytes, ependymal cells, oligodendrocytes, and then the microglia. Right, so you can see here, not that you need to know what they look like, but this is what an astrocyte would look like, an ependymal cell, an oligodendrocyte right here with these little extensions right here, and then the microglia. And we'll talk about what they do. Let's start with the astrocytes. So astrocytes have large cell bodies with many processes. Astro means star, right? So they kind of have like a star shape to them, and hence the name astrocyte. These have some important functions. Again, we're looking at neuroglia that are in the central nervous system first, right? These are only in the brain and the spinal cord. So one of the most important functions of the astrocytes, they maintain something known as the blood-brain barrier. I always abbreviate that to BBB. The blood-brain barrier is a highly selective barrier that regulates the transfer of solutes and chemicals between the circulatory system and the central nervous system. We just can't let anything get into the brain or spinal cord. It could be fatal if it's a toxin or something like that. So the astrocytes are kind of like the bouncer, right? They decide what goes in and out. The astrocytes also provide structural support. They regulate ion, nutrient, and dissolved gas concentration, right? They're just making sure we have proper amounts of sodium, potassium, calcium, that type of thing. They absorb and recycle neurotransmitters and even form scar tissue after some type of injury. So if you take a look over here, this is just showing you a blood vessel on the right-hand side. And then in blue, this is a axon of a neuron. And then here is the astrocyte. You can see it's even connecting like the blood vessel over and keeping it close to the axon over here. And you can see all these different processes. So this would be an example of the astrocyte. The second cell that we have are known as ependymal cells. So ependymal cells have an important job, right? They produce as well as monitor the cerebrospinal fluid, right? Cerebrospinal fluid is a fluid that actually bathes our brain and spinal cord, right? We're actually floating in water, the brain and spinal cord. So these are the cells that help to make the cerebrospinal fluid. They even have some cilia, remember the hair-like projections that help to circulate the CSF. So where do we find them? Since they're making cerebrospinal fluid, we actually find them in the ventricles of the brain. The ventricles of the brain are responsible for making the cerebrospinal fluid, and we also have them lining the central canal of the spinal cord. Let's take a look. So this is a cross section of the spinal cord right here. This little canal right in the middle of the gray commissure right there, that little canal is the central canal. So here it is blown up. So now you can see all along the central canal, or if this was a ventricle in the brain, we would have these ependymal cells right here. Right? And their job is to make cerebrospinal fluid. We'll talk about that in a separate lesson. The third cell that's in the central nervous system is known as the oligodendrocyte. Now, these have a small cell body, and then they have a couple processes extending off of them. Their job is very important. They form the myelin sheath. We're going to see in, again, a subsequent lesson, the job of the myelin sheath is to speed up the nerve transmission, right? They also make our neurons appear white. So this is what we would call white matter, right? They actually look very, very bright white. Now, the myelinated area on the neuron is known as the internode. But the oligodendrocytes don't touch one another. There's actually a space between oligodendrocytes, and then we have a bare area of the axon visible. The newer term is known as a node, right? We used to call it the node of Ranvier. These are the spaces between the internodes or the myelinated segment. So it would be something like this. Over here, let me make this just a little bit bigger for you. Over here, let's say we have an oligodendrocyte right there, and then you see it has a process, and what you can see here, the process is wrapped around the axon in blue. This is the axon, and the process is wrapped around over and over and over again, myelinating this piece of the axon. Okay, here is another process of that same oligodendrocyte myelinating the axon here. But notice this myelin down here is not touching this myelin over here. This gap right there is known as the node or the node of Ranvier. The part of the neuron that is myelinated, all of the axon in here that's inside, that's known as the internode, right? So that's the myelinated part of the axon. The bare area where the two oligodendrocytes don't touch, that is known as the node, right? They actually have it labeled over here, so that would be like right there between this cell and that cell. Again, this cell is cut open, so that's why it looks like that. Okay. So that's the node of Ranvier. And as I said before, the oligodendrocytes or the myelin that's on the axon appears white. So this is what we would call white matter. All right, so that's one of the major functions that I'd like you to know about the oligodendrocytes. They form the myelin sheath. Kind of a secondary function is they also provide structural framework, right? They kind of just support the whole brain and spinal cord. Okay, the fourth Neuroglia is known as microglia. This is more like a white blood cell. Right? These are the smallest and the least numerous of the neuroglia. They have many fine branch processes, and their job is to just kind of surveil, right? They kind of go around and they monitor the nervous tissue, and they will clean up any debris, any waste, or any even any pathogen, right? A pathogen is a disease-causing organism. So if there's a bacteria or virus, right? This is like the white blood cells. They actually are derived from monocytes, which you'll talk about in AMP2, which is a type of white blood cell. Okay, so let's take a look at the microglia. All right, so this was the astrocyte over here. Right? This was the ependymal cell. This little guy down here is the microglia. You can see these fine little branches coming off of it. Right? So this kind of just migrates around. Again, over here is an oligodendrocyte, just wrapping around the axon, producing the myelin. Okay, so that is our four neuroglia of the central nervous system. The final neuroglia are in the peripheral nervous system, so they're in the nerves. We have two. We have the satellite cell and we have the Schwann cell. Let's take a look at the satellite cell first. So we said in a previous lesson that the cell bodies of unipolar neurons, sensory neurons, don't live in the brain or spinal cord. They live just outside in these clusters known as ganglia. Well, surrounding or providing support to these cell bodies in the ganglia are a type of support cell known as satellite cells. Okay, so these surround the neuron cell bodies that are living in the ganglia. And again, I just put the definition one more time. A ganglia is a cluster of cell bodies in the peripheral nervous system. And their job basically is to really regulate the interstitial fluid around the neurons. So they're just kind of supporting those cell bodies in the ganglia. Let me just see real quick here, probably this best picture. Not that it's a great picture, but this is just showing you here a picture of a satellite cell. A little bit more importantly now, the Schwann cell is the cell that's equivalent to the oligodendrocyte, right? We just talked about the oligodendrocyte in the central nervous system providing myelin. Well, the Schwann cell is the equivalent cell in the peripheral nervous system. So listen, this is kind of an outdated term, but a lot of people still use it, so I left it in the notes. The newer term is known as the neurolemocyte. They form the myelin sheath around the axons, again, in the peripheral nervous system, in the PNS. Right? What will happen is a myelinating Schwann cell will just wrap around one portion of the axon. So it'd be something like this. If my finger was the axon and I have like a little flashlight here, let's say this is the Schwann cell. The Schwann cell will attach to the axon and then wrap around over and over and over again, up to 100 times it'll wrap around, insulating that area of the Schwann cell. So um, in order to myelinate an entire axon, we're going to need many Schwann cells. The oligodendrocyte was a little bit different. One oligodendrocyte had branches and it could do multiple axons, right? This is just one Schwann cell just doing one section of an axon. Let me show you. So let's kind of review from a previous lesson. Cell body, right? The cone area here, which is the beginning of the axon, is the axon hillock. Here's the initial segment. And then the blue right here is the axon, right there. But these structures here that look like hot dog buns, right? These are the Schwann cells. And the Schwann cells, I like this picture here because they cut the Schwann cell, you can see has wrapped around over and over and over again. Right? All of those lines represent the revolutions of the Schwann cell. All right, so each Schwann cell just covers a section of the axon. But notice the Schwann cells don't touch, right? There's a gap, there's a gap, there's a gap, right? Those are known as the node of Ranvier, right? The newer term is just a node, right? So just like we said with the oligodendrocyte, the parts of the oligodendrocyte that don't touch are the nodes. Okay, so this myelinated axon here, this would actually appear white as white matter because the myelin or the Schwann cells make it look white. Okay, and that is our neuroglia. Again, a total of 6 support cells, 4 in the central nervous system and 2 in the peripheral nervous system.